Okay. Another thing that I gave you are infinitely interesting. Go back. This is more interesting, I promise. Kind of. So we had a couple things to go over in peroxisomes. I decided for time and interest sake, especially since I'm going to take up. I spent like 13 minutes handing things back. I think over the course of this course, I've used up a class period handing things back to you. This is what not having TAs in the classroom does. So we're going to just move straight on to cytoskeleton, which we need to pound through. We're going to talk about how peroxisomes make light and things like fireflies and stuff like that. But that was cool, but this is more foundational. So let's go on and talk about the cytoskeleton. And really, as we get into this, there are three major parts of the cytoskeleton that you will need to know. Three major components I'm going to take you through and talk about. First off, we have these things called microtubules. We'll talk more about those in a few moments. Microfilaments. I think it gets split into two different words. There should be one word. And then also intermediate filaments. So they all kind of like the same name almost. I kind of feel like if people were really trying to make these confusing, this is about as good as you could do because, hey, you have micro in both of these and you have filaments in both of these, so that makes it nice and confusing. Okay, but let's talk about what the cytoskeleton does, why it's there, and really, you have two major functions or major domains of functions of the cytoskeleton. The first of these is kind of a static function, similar to the static function of your skeleton, which is really to act as a skeleton, to hold things in place, to hold the structure of the cell, the shape of the cell, and to do essentially the same thing that your skeleton does. So, again, we've talked about some of these functions as we've gone along the quarter by attaching things to the cytoskeleton. We're going to provide shape, support, organization. You may have heard me talking about we actually attach sometimes organelles to cytoskeleton to keep them in a certain place. Sometimes we attach enzymes to, or groups of enzymes into certain locations using the cytoskeleton so that we can arrange these things that we need into similar locations so that they're all doing the job in the same place. So, for instance, the products of one enzyme are in close proximity to the enzyme that needs those products as its substrate, and other things like that. However, this isn't the only kind of functionality that the cytoskeleton has. It also takes on quite a bit of dynamic function as well. This can generate movement, being one of them, and not just movement of the cell as a whole. We do talk about the cell as a whole moving based on changes in the cytoskeleton. And this is particularly more pronounced, more easily observable with unicellular organisms because you see them like amoebas and other things moving around in like pond water or something else like that. That's fairly obvious movement, which is being carried out by the cytoskeleton. We're also talking about movement that happens inside multicellular organisms. There can be movement of parts of the whole cell to change cell's shape or other things like this to adapt to a changing environment, changing neighborhood of other cells. We're also talking not only about movement of the whole cell, but movement within the cell as well. And so we need to generally move things within a cell to move it from here to there, or there to here, or other sorts of directions, because there and here is good directions. And we move them around to get them to where things need to go. We've talked already about vesicles moving around, and those need to go places. This is movement inside the cell, not movement of the whole cell, and the cytoskeleton plays a very important role in those things as well. So, I guess I just said all that. So, both for movement of the cell as a whole, and then also individual components inside the cell. Yeah. I didn't make myself. I'm sad. No tea.
If you stop the lecture, will you grab some tea?
No, it's okay. I'm behind. I'm like a day behind. 2 days behind. Maybe like a week behind. I don't know. I'm behind. I haven't imparted enough of my knowledge into you as I'm supposed to. I'm a failure. I get used to it. So let's first talk about microtubules.
We need failure.
Yeah. What's that? Okay, so that's flattering. Structure and assembly. Okay, so let's talk about microtubules. This is the first of those major components that we're going to talk about. And first off, these are made up of this protein called tubulin. Kind of makes sense. We have microtubules, and it's made up of this thing called tubulin, because not only are we good as biologists at making confusing names, we're also sometimes fairly unoriginal. So tubulin is a protein heterodimer, meaning that we have two different polypeptides that make up the entire protein. Hetero meaning that they're two different, they're not the same subunit. We have an alpha and we have a beta. And we also refer to these in polarity-wise as the beta side as the plus side and the minus side as the alpha side. And this is going to come up a little bit more in as we go on and look at how this creates an entire microtubule, but keep this in mind. The alpha side of the tubulin is the minus side, the beta side is the plus side. This is a fairly large molecule actually, so tubulin is like about 110 kilodaltons, it's fairly big. And actually in you have a lot of several different kinds of tubulin. We have 6 types of alpha and 6 types of beta, although at this moment we're not exactly sure why we have 6 different types. They don't seem to be all that much different, they're different. Now, we can take each of these pairs or these tubulin dimers and we can string a bunch of them together and get this. So we can get a polymerization of these dimers, and this would be known as a protofilament. Which makes sense. We're in the process of making a filament, so a string of these dimers would then be a protofilament. If we want to make a whole microtubule, we take 13 of these protofilaments and we arrange them in a tubular fashion, hence the name, I guess. And we have a hollow core in the middle, and that is a microtubule. So it looks something like this. We have 13 of these protofilaments around, and so actually we're making up lots and lots and lots of the individual tubulin dimers. It's arranged in a kind of spirally sort of shape here, in that if you look at the alpha subunits, the next one adjacent to it, or the protofilament adjacent to it, is going to have its slightly advanced, and slightly advanced in the minus direction. If you're going one way, I guess it's going the other way. Slightly unadvanced, slightly behind. These are actually relatively big. So the entire microtubule is a structure that's about 25 nanometers in diameter, and they can actually be several millimeters in length. So this, for individual molecules, or an individual, like, protein here, this is a fairly long thing. In fact, ginormous. Okay. So let's talk a little bit about this plus and minus end then. So we have, therefore, a plus, the beta end, and the minus end. And hopefully this makes sense why they're called this, because it actually turns out that as we polymerize microtubules, the plus end preferentially grows. It's growing about, as it's polymerizing, you're polymerizing about twice as fast at the plus end than you are at the minus end. And this is for both assembly and disassembly. So as we're polymerizing or depolymerizing, is that a word? Delimerizing? No. As you're taking it apart, most of that's preferentially happening, or about twice as much is happening at the plus end. Hence the name. Okay, so I mean, essentially like this. For every 2 that we add here to the plus end, we're going to add on 1 to the minus end as we're polymerizing this microtubule. Okay, so that is a single microtubule, but they don't always necessarily just occur singly as one single microtubule. Looking at these kind of end-ons, or we're looking at the entire ring from the hollow core end, what I just showed you was a singlet, so you can have a single microtubule. But we can also have these occur in doublet, where they're sharing between each of the microtubules 3 of those protofilaments are being shared between each of the microtubules, and you get doublet. Or we can additionally have a triplet as well, where we have 3 microtubules, essentially 3 independent microtubules, each pairwise are sharing, again, 3 protofilaments between them. So these aren't necessarily, oftentimes, as we are constructing microtubules, we want them to have some sort of organization. We're not wanting to have them just assemble just randomly out in the middle of nowhere without direction or anything else like that. So, especially in times where we want to have a high amount of organization, we have these structures associated with that, which are MTOCs, or microtubule organizing centers. And this is a structure that essentially exists long term, but you will have microtubules being constructed from this. So essentially, this is almost like a nucleation center for other sorts of, if we were to refer to it in other terms. It's a beginning of a microtubule that will then add on additional tubulin onto here to extend the microtubule from. So a couple examples of this is centriole in cells. These are a point where we get microtubules that we are going to be using for cell division, and particularly organizing the DNA as we go through cell division. And this is a hollow tube of 9 triplets of microtubules, and they're fairly short. We'll look at that in just a second. And again, these are making up kind of inside the centrosome. This is what it becomes as the centriole actually engages in cell division.
So the centriole, or the centrosome is a centriole.
Yes. Okay, so if we look at this, this is essentially a centriole right here. What you have here is kind of a little bit hard to see, but we have these triplets of, or these microtubule triplets, and then we have 9 of them, 1, 2, 3, 4, 5, 6, 7, 8, 9, surrounding each other, or kind of making up a hollow tube. And then these are going to be the sites at which we can begin to polymerize microtubules and maintain a high degree of organization when we decide we want to create those microtubules. Any questions on that? We additionally have these other things called MAPT, or microtubule-associated proteins. So oftentimes these microtubules have to be doing something. They're carrying out some function. And the microtubule structure itself provides structure, but it itself isn't going to really do anything. We need these microtubule-associated proteins, they're just accessory proteins to those proteins, to carry out many of the specific functions that that microtubule needs to do. We'll come back and talk more about some of these in just a few moments. And really, it can end up taking up a large amount of when we actually pull out microtubules, if you try to isolate them and their associated proteins, it turns out about 20 to 30% of the microtubule, when you take out the entire, everything as associated proteins, about 20 to 30% is these MAPs. So a good amount of associated proteins are with these microtubules. Okay, so let's talk about a couple different examples of the uses of microtubules, and one of the really kind of nice and obvious ones where we're doing a kind of fairly dynamic function is a eukaryotic flagella, and also cilia, but we'll talk mostly about the flagella. So flagella, these are extensions off of a cell. They tend to be fairly long, to about 150 micrometers, and they tend to be small in number. You tend to not have many flagella off of a single cell. It can be anywhere from 1 to 10. I mean, a lot of cells have no flagella, but if you have a flagella, it'd be anywhere from 1 to 10. And the purpose of a flagella is to propel a cell through liquid, generally to propel a cell through liquid parallel to the flagellar axis. And so if you have the well, let me show you here real fast. Here's an example of like one flagella and the direction. So here's the flagellar axis right here. And so the direction of propulsion would be kind of this way. Same with these. You can sometimes have many flagella all come out the same side. Sometimes you can have flagella come out both. Sometimes you can have like crazy Medusa hair of flagella as well. Then we also have cilia. Cilia are somewhat similar, especially structurally, to flagella, but in terms of cilia, however, they tend to be shorter, only 5 to 10 micrometers, and we tend to have very, very many cilia. So cilia can be anywhere in the hundreds to the thousands, and cilia are not always used for propulsion, but when they are, they tend to propel liquid across the cell surface, and so the cell itself could be stationary and the cilia be propelling liquid, or the cell could be trying to propel itself through liquid. But when it does this, it is tending to move the liquid perpendicular to the axis. So, if we were to take a look at this, you have a cell, you have cilia off of it. This is a happy cell, so we have a there. It's really weird at the moment. There we go. Nose. Anyway, so these cilia, these are not hair, these are cilia. It will tend to beat this way, and we're going to tend to move water or the liquid that way. That really, okay, so that just ruined it. It's a neutral cell. Okay, so those are cilia, or kind of a buzz cut, I guess. So let's talk about, so really why I'm telling you about this is because of their structure and the use of microtubules in these structures. And so let's talk just a moment about this. In flagella and cilia, we have this common structure where we have two central microtubules that run down the length of the flagella or the cilia. And these will be surrounded by 9 microtubule pairs. These are doublets surrounded by a plasma membrane. So the entire structure then is surrounded by a plasma membrane. So we kind of have this structure where we have a 9 plus 2, the 2 being the 2 central microtubules, the 9 being the 9 doublets that we have that surround the periphery. We can also kind of say that we have alpha and beta tubules. I'll come back and talk about this in just a few moments more, but in each of these doublets, the two microtubules that make up the doublets are not the same. They have slightly different structures and slightly different functions. And so we call them A and B tubules. Did I say alpha and beta the first time?
Yep.
Okay, I meant A and B, excuse me. Alpha and beta tubulin, A and B tubules. That make up the doublet. So let me show you what that looks like. We have two microtubules here that make up the center, and those are running, so this is kind of a cross-section of a ciliary flagella. We have the two microtubules here that make up the center. Then we have these 9 doublets around the exterior, and in each of the doublets here, again, we have a beta, or a B tubule and an A tubule that, again, as you can kind of see here at the moment, they have slightly different structure. We'll come back to those here in just a minute. So that's the basic microtubule structure of the cilia or the flagella, but we also have several maps that are associated with that as well, which that was kind of repetitive. We have several microtubule associated proteins. The first one are these radial spokes. These are proteins that are connecting the microtubules of the sheath to the central portion of the cilia or the flagella. Now, they're helping to control the coordination of movement of the entire flagella or cilia, but we're not exactly sure how they do that. So let's take a look at what around here is this. So these right here are the radial spokes. These proteins extending from the A, not alpha, A tubule in towards the center here. And we also have another map called nexin. This is going to connect adjacent pairs of the doublets around the periphery, but then also connecting the central pair as well. So nexin is these right here. It's in pink. This protein connecting the adjacent doublets and then also connecting the central pair as well.
Can you go back?
To that? Okay. Okay. Then we also have, and this is one of the most kind of dynamic of the maps associated in the flagella or cilia, is dining. Dining is a molecular motor, which we're going to talk a little bit more about in just a few moments. We might get to it in this class period. That allows for the movement of the cilia. So we have them actually attached to the alpha tubules.
The A tubule?
To the A tubule, thank you. See, we named these really confusingly. I want to talk to whoever named these and be like, that's not really talking, I guess that's just sighing really loudly. And it might be long enough to go there to be like, what? I say, could you please name, okay, whatever you do, not microtubule, microfilament, intermediate, those are just way too similar. And things in my brain get way too, and then alpha and beta and then A and B, no. Especially when like alpha and beta have a plus and minus, like if you just call them plus and minus and like forget the alpha and beta and then call these A and B, I would be happy. But you know what, it's not even probably a single person either. It's like a conglomeration of people just like together, like doing stupid things separate from each other, but like the net culmination is just less than ideal. And you'd think that like a central authority could like figure this out and like make it better, but it doesn't help anything because like taxonomy has the same thing where a central authority like decides, okay, these are good names and these are bad names, and you still have stupid stuff happening in taxonomy. So okay, that's my little rant for the moment. So anyhow, going back to dining. So the dining's really cool because it's permanently attached to the A tubule. However, the other side of it can reversibly bind to every 10th subunit of the beta on the adjacent pair. So this is not on the pair of itself, it's attaching to the adjacent pair, and then what it will do is it'll essentially ratchet. Now I have that here? And then we have this ratchet-like motion that will essentially pull itself along the beta tubule, or the B tubule, and this requires ATP, and what this does is, as you get one side moving relative to the adjacent ones, you get bending of the entire flagella. Or depending on how all 9 of them are working together, you can also get a twisting or kind of a spinning motion or something else like this, depending on how we actually get that working. So we get each of these moving and sliding somewhat independently of each other, but in a coordinated fashion. So they're not all moving at the exact same time. So we get some sort of coordinated movement based on how each pair is sliding relative to the pair next to it. I'll hold that just for a moment. Oh, whoa. Back here just for a second. Wait, go back there! Sorry. It's sometimes fine if I make odd voices at my technology, it works better. Or maybe it just like eases my mind so it feels like it works better, I don't know. Okay, so to take a look at what this looks like, here's the dining right here, and these will attach and ratchet up on these tubules like this. They kind of walk. So this will bind to this B tubule from this alpha tubule, or A tubule right here, and then they will like that. We'll talk more about that in a little bit. This is actually a scanning electron microscope of a cross section of cilia here, I believe, and so you can kind of see. Yeah, it actually, like, in reality, it actually looks like that. So again, we have our doublets here. We have 9 doublets around. We have the centers, or the 2 center microtubules there. You can kind of see the dining a little bit here, and maybe like a little bit there. Or not the dining, excuse me, the nexon. The nexon is more, shows up there. Okay. Okay, so that's the actual structure of the main trunk of the cilia of the flagella itself, but it needs to be anchored into the cell by something, and that is what's known as the basal body. The basal body is a microtubule organizing center. So this is an example of one of those MTOCs that we were talking about a little while ago. That's not a centered sum. Now, it's slightly different. Instead of having 9 doublets surrounding 2 central, we only have 9 triplets and no central microtubules in the basal body. We do, however, have the radial spokes moving inward, and we also still have the nexon that is connecting the adjacent triplets in this case. However, we have no dining. This is just an anchoring structure, so it doesn't. These microtubules don't need to move relative to each other, so it makes sense we have no dining here. Let's take a look at what those look like right here. So again, we have a flagella here. Once again, we have the 9 doublets surrounding 2 centrals. We have nexon in between here. It's showing the radial spokes going in here, and then we have the dining. Wait, did I say dining already? And the radial spokes, nexon, and then we have the dining here that's going to go like this and cause that. Again, we have all of this is being surrounded by the bilipid membrane here, and you can see it kind of going smoothly into the rest of the cell. And then down here we have the basal body. And looking again at what this looks like, we have no central microtubules, but we do have the 9 triplets around here. And then we have nexin connecting each of these triplets. We have radial spokes, that's not shown here, moving in towards the center, but we don't have the dynein. Oh, go back there. Go back! Back, I said! Okay. There. Yeah, skipped all that. There we go. That one? Yeah. That one. And we're going back here. Okay, so from there I'm going to skip a couple more and we're going to go through this. Okay, so let's talk a little bit about these molecular modes just for a few minutes before I let you out here. And these are particularly proteins that are useful for movement. We're going to use them to ship things around the cell, and we're going to use them on the microtubule. So these are microtubule associated proteins. So one particular ones that we have is things like kinesins. These are going to walk along the microtubule, and it really kind of is like that. And they're going to walk from the minus end to the plus end. That's going to be the tendency of them to walk. They actually can sometimes take steps backwards, but the most of their steps are going to be from minus to plus. And actually, this is not one protein. There's actually a superfamily of different kinesins or kinesins, depending on who you're talking about. Kinesins out there, they look a little bit like this. Oh, hey. That's, okay, so we have 2 different parts, and this is upside down. So down here, we have feet down there that attach to the microtubule. Kind of 2 major domains. Each of these are ATPases, so they use ATP to actually go through and do each step of their walk. And then there's 2 long arms that are intertwined, and then we have 2 sections at the other end that we can bind things to that we want. I'm going to go very briefly through how this works. So again, those 2 major domains are going to attach to the microtubule. Each of them binds to the tubulin. And it happens kind of like this. We're going to have ADP attached to the feet. As it has ADP attached to it, then it can bind to one of the beta subunits, and it's going to allow it to then bind to ATP. And then the other foot does that as well. It is then going to hydrolyze the ATP to ADP, which then allows it to unbind and then swing forward, and then bind again. And that's going to be replaced with ATP. So let me look really fast, do this in motion, and see if that makes more sense here. Okay, so this head has ADP on it. It binds to one of the beta tubulins. And then the ADP is going to be replaced by an ATP, which allows it to unbind. But it's not going to unbind quite yet, because I'm going to show you that the other one also binds. So the ADP then, now that we have that phosphorylated, it unbinds, or that we hydrolyze the ATP, and then it can swing forward. And then again, it's going to bind, and we're going to replace the ADP with the ATP. Meanwhile, the other one hydrolyzes its ATP, which allows it to unbind and swing forward as well. I like this animation because it kind of, as you see them, it makes it look like animations of this. It makes it look like a very, like, it's just like walking very purposefully. But in the end, actually what's happening is a very kind of stochastic process where it unbinds and then the arm's kind of like swinging around. And it will more often than not bind again in the plus direction and then rebind. However, it does sometimes bind further back and then, yeah. But more often than not, it will bind forward. So animations make it look a little bit something like this often. You have the kinesin that will bind and take its steps along our microtubule. On the other end, we're going to have a vesicle with a kinesin receptor on it that will bind to the other end of the kinesin, and then we can move this entire vesicle somewhere based using this motor, as long as the microtubule is going in the right direction from plus to minus. Okay. Here's a couple more real quick things. Dynes do kind of, yes, Monica.
Back to that last picture, did the vesicle bind to the kinesin receptor on the kinesin, or does like the vesicle and kinesin receptor go bind to the kinesin?
The vesicle is on, or the kinesin receptor is on the vesicle.
So it never leaves in the vesicle?
Yes, and then the vesicle, well, no, it never leaves the vesicle. And so you would see this part kind of around, and then this part without the receptor, and then the vesicle will come, kinesin receptor plus vesicle would bind to the kinesin. Does that make sense? Okay, yeah. Make sure I'm communicating with this correctly. Okay, here we have dyneins. They move in the other direction from the plus to the minus end, opposite of the kinesin. However, both of these use ATP. So essentially we're using ATP to drive each step on both of these. And they don't go very fast. They're going like 1 to 2 micrometers per second, but in the grand scheme of things, this means within like a minute they can get just about anywhere in the cell they need to. So again, cells usually aren't more than 100 micrometers max, and so in a minute and a half you can get anywhere in the cell. And they produce about 1.9 piconewtons per molecule, and each step size is about 8 nanometers. Are there any questions on that?
So are these ATPases too?
Yes, both dyneins and ATP kinesins are. Well, with that, I will see you on Wednesday. Enjoy your dead week, by the way. Hey, it's dead week. Dead week happens. It's happening. Like in the process of happening. I won't give you a surprise test this week. No, this is how the G protein. The G protein is moving more randomly within the cell. Yeah. So it's not moving by. As far as I know, it's not moving by. I don't know.
What was that? So, is minus moving? Is it renewable phosphate?
No, it's an ATP bonds, and then the ATP